Cellular respiration. Cellular respiration is a multi-step metabolic pathway whereby organic molecules, glucose, fatty acids, amino acids, are disassembled in a controlled manner, regulating blood levels of nutrients. The regulation of blood nutrients is dependent upon the length of time since you have eaten. So we'll start off with the absorptive state, also known as the fed state. The fed state includes the time you are eating, digesting, and absorbing nutrients. The fed state usually lasts about 4 hours after a meal, and during the fed state, the concentration of glucose, triglycerides, and amino acids increase within the blood as they're being absorbed inside the GI tract. The body must maintain this homeostatic level of nutrients in the blood while absorbing nutrients. Insulin is a major regulatory hormone that is released during that fed state. The release of insulin from the pancreas occurs in response because there's higher amounts of blood glucose levels. The liver will convert excess glucose into glycogen. The muscle will take excess glucose, convert that into glycogen, and amino acids could be used to build contractile proteins. Let's not forget, adipose tissue will also store excess lipids as well. The post-absorptive state is also known as the fasting state. And the fasting state is the time between meals when the body relies on the storage nutrients because there's no further absorption of nutrients occurring. Glucagon is going to be the major regulatory hormone that is released during the fasting state. The liver goes through glycogenolysis and lipolysis, leading to the release of glucose and lipids into the blood. If you don't eat, the muscle will also break down, leading to release of amino acids and glucose being released into the blood, and of course releasing some of the stored lipids. The glucagon promotes the release of glucose from the liver and the release of fatty acids and glycerol from all the storage sites. So it's the exact opposite of the fed state. Now let's talk a little bit about some of the enzymes in cellular respiration. There are several types of enzymes that we're going to cover. Even though there are thousands of enzymes, they have been classified into 6 major categories. We have the oxyreductase, we have the transferase, we have the hydrolase, we have the mutase, dehydrogenases, and lyases. The hydrolase is an enzyme that cleaves one molecule by breaking down a covalent bond. A mutase, also known as isomerase, is an enzyme that will catalyze rearrangement of an atom. A dehydrogenase is an enzyme that will remove the hydrogen atoms, usually protons, from a substrate, and therefore transferring and moving electrons. Transferases are going to catalyze the transfer of a functional group, a phosphate group, a methyl group, from one molecule to another. An oxyreductase is going to catalyze the transfer of electrons involving oxidation of one substrate and the reduction of another, or the loss of electron on one to the gain of electron to another. Lyases are going to catalyze the breakdown of a covalent bond without any water or oxidation. On the right, we have several examples of these kinds of enzymes. Just don't forget, anytime you see the word that ends in the 3 letters of a suffix ase, that means it's going to be a subclass of an enzyme. Glycolysis, which is anaerobic cellular respiration, is the first stage that occurs inside a cytosol and is anaerobic, and that transforms glucose into 2 pyruvate molecules plus 2 ATP plus 2 NADH. So let's take a look at each of these steps and each of these key enzymes and intermediates. The first step begins with glucose being converted into glucose 6-phosphate with an enzyme called hexokinase. It attaches phosphate from ATP onto the phosphate. The second reaction happens with the enzyme glucose 6-phosphate isomerase that will then take the glucose 6-phosphate and rearrange it into fructose 6-phosphate. At this point, we're still working with 6-carbon molecule. Then the third step involves an enzyme called phosphofructokinase. This will then take the fructose 6-phosphate and rearrange that with 2 more phosphates in creating fructose 1,6-bisphosphate. This reaction is not shown in the image. Then step 4 utilizes another enzyme called aldolase to take that fructose 1,6-bisphosphate and convert that into a glyceraldehyde 3-phosphate. This is where we split the 6-carbon molecule into 2 3-carbons. So moving forward, every single reaction down here happens twice. This is because it is now 2 3-carbon molecules instead of 1 6-carbon molecule. Step number 5 utilizes an enzyme called glyceraldehyde 3-phosphate dehydrogenase that will then create 2 NADH and rearrange G3P into 1,3-bisphosphoglycerate. Notice that there are 2 phosphates attached to the 3-carbon molecule. The 6th step is going to be the phosphoglycerate kinase, which will then take the phosphate and attach it onto ADP to create the first 2 amount of ATP in the reaction. Then step number 7, we use an enzyme called phosphoglycerate mutase, which will take 3-phosphoglycerate and convert that into 2-phosphoglycerate. This simply rearranges the phosphate location on the carbon. At this step, you'll see that we would lose a water molecule. Moving to step 8, we use an enzyme called enolase, which converts the reaction of 2-phosphoglycerate into a phosphoenopyruvate. And then phosphoenopyruvate gets converted into pyruvate with the last enzyme called pyruvate kinase, PK. So at the end, there is a net gain of 2 ATP and 2 NADH molecules. So glycolysis is regulated by a negative feedback process in which ATP acts as an allosteric inhibitor, and it can be used to turn off steps inside this reaction. The main enzyme that gets targeted is step 3, phosphofructokinase. This enzyme is what responds to an increase in ATP and turns off to stop glycolysis. So when pyruvate is made, there's 2 options. The first option is if oxygen is available and sufficient, pyruvate can enter the cell's mitochondria and complete aerobic breakdown, yielding water and CO2. And if there's not sufficient amount of oxygen, then pyruvate can be converted into lactic acid. This next step is called the intermediate stage. The intermediate stage, along with the citric acid cycle and the electron transfer chain, all occur inside the mitochondria. The mitochondria is a double membrane organelle that has an outer and inner membrane, and it has these inward folds called the crista. The fluid-filled space between the 2 membranes is the outer compartment, and that innermost space is called the matrix. During this intermediate phase, an enzyme called pyruvate dehydrogenase brings together pyruvate and a molecule of coenzyme, also known as CoA, that is already present within the matrix to form a compound called acetyl-coenzyme A or acetyl-CoA. In this reaction, it removes 1 carbon, giving off some CO2, and it will also create NADH with the proton and electron on it. So the final product of the intermediate phase is this carrier that helps transport the acetyl group, also known as acetyl-coenzyme A. This will then get shuttled into the Krebs cycle for further production. The resultant is acetyl-CoA from the intermediate stage, which now can enter the citric acid cycle, or the Krebs cycle, which also occurs inside the cell's mitochondrial matrix. Let's not forget that the intermediate stage must occur twice for each glucose molecule. 1 glucose molecule yields 2 acetyl-CoA. The citric acid cycle, also known as a Krebs cycle, is a metabolic pathway that occurs through an activity of 9 enzymes located within the matrix of the mitochondria. Some of the older texts call it the tricarboxylic acid cycle, but during the citric acid cycle, the acetyl-CoA that was produced in that intermediate stage is now converted to 2 carbon dioxide molecules, and the coenzyme molecule is released. The energy is released as a result of the citric acid cycle. And this energy is used to form 3 molecules of NADH, 1 molecule of FADH2, and 1 ATP molecule for every 1 turn of the cycle. There are 2 turns for each citric acid cycle because during glycolysis, the glucose was split into 2 molecules, creating 2 pyruvates. So let's take a look at each step. Step 1, the citric acid cycle, involves combining the acetyl-CoA from the intermediate stage with a molecule called oxaloacetic acid to form citric acid. The molecule then gives the citric acid its name. Step number 1, the enzyme that's involved is called citrate synthase. That takes acetyl-CoA plus oxaloacetate. Step 2 and 3 of the citric acid cycle form an isomer by removing the water molecule from citrate and then reattaching it on different parts of the molecule. Then you release the water and you use a second enzyme called aconitase to create isocitrate. Steps 4 of the citric acid cycle occur through different hydrogenase enzymes that participate in the transfer of hydrogen to NAD positive to form NADH. So the isocitrate is created into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and then alpha-ketoglutarate gets converted into succinyl-CoA with the 4th enzyme called alpha-ketoglutarate dehydrogenase. In these 2 steps, you'll notice that NADH is created and given off. Succinyl-CoA becomes succinate with the help of an enzyme called succinyl-CoA synthetase, and the ADP plus pyrophosphate becomes ATP. In this case, it's GTP. Then in step number 7, the citric acid cycle occurs through the action of another dehydrogenase enzyme that transfers hydrogen to the FAD to form FADH2. In this picture, it's expressed as Q. In this step here, we create FADH2 with an enzyme called succinate dehydrogenase that then creates fumarate from succinate. Then step number 8, we remove another molecule of water with an enzyme called fumarase, which converts fumarate into malate. And step number 9 of the citric acid cycle, when the oxaloacetic acid is again regenerated, and the cycle starts again with an enzyme called malate dehydrogenase, creating another NADH, and the malate becomes oxaloacetate. So the citric acid cycle is regulated so that there's not an overproduction of energy or an underproduction. The regulation of the citric acid cycle primarily occurs at the enzyme in the first step, which is the citric synthase. This is the most important step regulating the speed and the availability of this process. So in summary, the glycolysis is an anaerobic process, whereas the Krebs cycle is going to be an aerobic process. And during the Krebs cycle, this occurs inside the mitochondria, and its main focus is to produce acetyl-CoA molecule, CO2, NADH for every one turn. So in summary, the aerobic citric acid cycle occurs inside the matrix of the mitochondria and produces two CO2 molecules, 1 and 2. It produces one ATP molecule, or GTP over here, one FADH2 molecule right here, and a total of 3 NADH, 1, 2, and 3. All the enzymes are found inside the matrix of the mitochondria except for succinate dehydrogenase. This enzyme right here is the only one that's found inside the mitochondrial membrane. If ATP is needed, it will then go into citric acid cycle mode. If ATP is sufficient, then fatty acid synthesis could occur. There are some conditions that could affect the cycle. Poor nutrition, hypoglycemia, or even drinking a lot of alcohol can cause more lactate to be created from pyruvate and also more malate to be created from oxaloacetate. And that could affect the way the metabolism works. There's a mnemonic that I'd like to share called citrate is Krebs starting substrate for making oxaloacetate. Citrate is C, I is isocitrate, K is alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. The electron transfer chain involves the transfer of electrons or energy from coenzymes NADH, FADH2 that were produced in the first 3 stages of cellular respiration, from glycolysis and the Krebs cycle. The electrons or the energy from FADH2 and NADH are used to produce ATP. There are several significant types of molecules that are embedded within the crista of the mitochondria, and these are called electron carriers, hydrogen protein pumps, and ATP synthase enzymes. This is where the ADP goes through the enzyme ATP synthase to create ATP. And the final electron acceptor is going to be oxygen, therefore creating a lot of water inside our cells. So let's take a look at the reaction of the ETC. In step number 1, NADH and FADH2 are oxidized by releasing hydrogen, electrons, and protons that are passed through a series of electron carriers in the ETC, eventually making its way to oxygen, the final electron acceptor. Step 1, the oxygen combines with 4 electrons, 4 protons, to produce 2 molecules of H2O. Step number 2, the potential energy is converted into kinetic energy, and electrons are passed from one carrier to another. Step number 3, the protons are transported through the ATP synthase, releasing sufficient energy to then form ATP from adenine diphosphate and pyrophosphate to make ATP. This process of forming ATP is referred to as oxidative phosphorylation. The number of ATP generated when electrons are released from coenzymes depends on the entry point of electrons in the ETC. The electrons from NADH can enter at the top part of the ETC and pass through the 3 proton pumps, which will then eventually release 3 ATP, or electrons from the FADH2 can enter the second pump directly, resulting in the generation of 2 ATP. So this depends on wherever the electrons enter in, whether it be from the first complex or the second complex. So let's talk a little bit about each of these complexes. So the first complex is where the 2 electrons are moved from NADH into the complex, and this is where it can pump up to 4 hydrogen ions across the membrane, right here. Complex number 2 is a quinone complex. Ubiquinone is a lipid-based complex that can directly receive and bind FADH2, and this does not interact with complex number 1. Complex number 3 is composed of a cytochrome B complex, and cytochrome B contains an iron bound to a sulfur protein. This is also the enzyme cytochrome oxyreductase. Complex number 4 contains 2 heme groups and also copper ion. This is why it can bind oxygen very tightly. So this is shown over here and on the bottom. Metabolism of fatty acids by beta oxidation. So fatty acids can also get broken down into energy, and this then eventually becomes acetyl-CoA. But how does it do that? Well, fatty acids with lots of carbons and hydrogens go through several steps. The first step in fatty acid activation involves converting fatty acid into fatty acyl-CoA. Fatty acyl-CoA is created by an enzyme called fatty acyl-CoA synthetase, and this occurs inside the cytoplasm. This will then be transported into the mitochondria via these carnitine transporters, and they form the fatty acylcarnitine. There's an enzyme called a CAT1 carnitine acyltransferase, which moves the fatty acylcarnitine into the mitochondria. Then the final step is going to be beta oxidation. This is where oxidation occurs that produces FADH2 and NADH, and there's going to be a theolytic cleavage that releases acetyl-CoA by snipping parts of the bond, releasing acetyl-CoA for your body to use and make energy. Now acetyl-CoA can go back into the Krebs cycle and then the ETC. Proteins can also be used for fuel when they are broken down into amino acids, and these amino acids are then deaminated, where the NH2 group are then removed. When these NH2 groups are removed, they can go into another cycle called the urea cycle, and then the pyruvate is going to be then converted into acetyl-CoA and goes right back into the Krebs cycle for more energy production. The continuation of glycolysis under constant anaerobic conditions. When there is no oxygen, the glucose, 6-carbon molecule, can keep on going, and because there is no oxygen, it can also go through fermentation. So once you get to pyruvic acid, if there is no oxygen available, it has the route to go into lactic acid, where the NADH is going to be converted into NAD positive, and this will then allow the continuation of more glycolysis to occur. Lactate dehydrogenase is the enzyme that converts pyruvate to lactic acid, and this builds up, causing muscle fatigue.